Hello, friends, welcome to our new channel, the Learning Hub. So, this is SEM 6 Industrial Microbiology, and in Industrial Microbiology, the first topic is Introduction to Industrial Microbiology. So, if you get any question related to industrial microbiology or any topic in this unit, so first you have to give the introduction about the industrial microbiology. So in these I will be explaining about the what are microorganisms, what is industrial microbiology, what is its scope, and microorganisms in industries, what is fermentation, production. All these are the production like steroids, vitamins, amino acids, organic acids, antibiotic, wine, vaccine. And also we will see the applications in industrial microbiology and also the future. Now, this topic, what are microorganisms? Microorganisms are nothing but these are indispensable components of ecosystem. What is meaning by indispensable components? This is nothing but without microorganisms there is nothing. Okay, they play an important role in organic and inorganic materials through their roles in carbon, nitrogen, and sulfur cycle. Also they maintain a stability of the biosphere. Okay, and also they are the source of nutrients at all the ecotropical food chain levels and all web. Okay, so all other forms also are dependent on these microorganisms. So, by this, we get to know these microorganisms are very crucial in our day to day life. Okay, now these are my own notes. Okay, you will never find these type of easy notes. Okay, now what is industrial microbiology? Now we have seen what are microorganisms. Okay, now let's see what is industrial microbiology. Now industrial microbiology is nothing but it is a branch of applied microbiology. Okay, this will be referring to the microorganisms which will be using in the commercial enterprises. Okay, like commercially if you use in any business, that type of microorganisms. Now these microorganisms are should be processed under, there should be controlled temperature, pH, and also uh, there should be everything controlled to produce valuable products. There is many scope for this microbiology, like there is food and dairy microbiology, industrial microbiology, genetic engineering, investigation microbiology, medical microbiology, immunology, and agriculture microbiology. This is, if you want you can write or else you can skip. Okay, now let's see what microorganisms in industries. Okay, so you have to remember here 7 points, like what is the main role in the industries. You can say it is a, okay, you can remember in easy way. Okay, in exam you can say that it is a mini chemical factory and it possesses a broad variety of enzymes, and enzymes are very important for our life. Okay, and it plays it's highly metabolic activity, and this can grow and multiply rapidly. Okay, these microorganisms have high metabolic activity, you can remember, and large surface area for the absorption of nutrients, and these will be growing on the inexpensive substrate. Okay, byproduct. So, and this is also amenable to genetic manipulation. Okay, it is easily acceptable, and it should not be pathogenic to humans and animals. So if you see, there are many microorganisms that will be harmful to humans, but whatever the microorganisms we are using in industries, it should not be harmful. Pathogenic is nothing but harmful. Okay, it should not be harmful to human and animals. 7 points. First one, it is a mini chemical factory. It possesses a variety of enzymes, and you can say high metabolic activity, can grow rapidly, large surface area. Okay, you need to by heart the answer. You can remember the main key points. Okay, it is grown in expensive substrate, amenable to genetic manipulation, and also it should not be pathogenic to human and animals. So this was about the microorganisms in industry. Now if you are talking about the industrial microbiology, every microorganism, okay, the fermentation is very important. What is fermentation? Fermentation is nothing but chemical conversion of sugar to acid or alcohol. Okay, and now this fermentation will be done in the large vessel. This large vessel is nothing but bioreactor. Also, we can call it as fermenter. So here the microbes will be growing. In these, there will be a controlled temperature, pressure, foam control. In later videos, I will be explaining a detailed video on this design of a fermenter, which will be very important for your examination. Okay, now just you can also write this point only for to make your answer valuable. Okay, science of fermentation is called as zymology. You can include this. Okay, now let's see the productions. We have seen this, many productions. Like first production is steroids. What are steroids? Steroids are nothing but these are man-made chemicals. Okay, like it's present naturally in the human body, like it's hormones. Okay, which play an important role in our human body. Now this is involved in the regulation of sexuality. Okay, first point you can write this. And now you have to write what are the microorganisms involved. Okay, make your answer easy. Okay, now rhizopus, nigerians, aspergillus, so these three microorganisms are very easy. Okay, you can remember this and you can write it in your examination. So these species are used in steroid production. Now, we can also say that microbial biotransformation. So it includes hydrogenation, dehydrogenation, oxidation, or this addition of side chain. This is also an application. Okay. Now vitamins. Okay, it is also these microorganisms are also useful for the production of vitamins. There are many types of vitamins like vitamin A, D, E, K that are fat soluble, and also B, B12, B6. You know that. Okay. So these are commercially produced by using microorganisms. So we can say that microorganisms can also produce vitamins. Now here two microorganisms are there, important, like streptomyces and acetobacter. So they can anaerobically. What is meaning by anaerobically is nothing but absence of oxygen, and aerobically is nothing but presence of oxygen. So they can produce an absence of oxygen, also in the presence of oxygen. So this is also called as cyanocobalamin. Cyanocobalamin is also known as vitamin B12. It is also known as vitamin B12. Okay. So this was only you have to write 3 points in production. Okay. Remember that. First, what it is used, किसमें use हो रहा है, and second, what are the microorganisms involved and who discovered it. So you can remember George Whipples. By writing this, the examiner can easily correct your answer and you can score the marks. Okay. Now let's see. So major sources what for vitamins? Egg, meat, and poultry. The third one is the production of amino acids. Here three major amino acids have been given. What are that? L. You can remember these three amino acids which are very important, which are industrially produced by these microorganisms. L-lysine, L-glutamic acid, and DL-methionine. So where in which fermentation it is produced? Means in which fermenter? What is the type of fermenter? Later videos I'll be explaining about the types of fermenter, what type of fermenter, in which type of fermenter what products are produced. Okay, now in this video let's only remember that in these amino acids are produced in the submerged aerobic fermenter. Okay, and it can be batch or fed batch. Okay, remember this 3 points. Okay, three amino acids, submerged aerobic fermenter, and batch or fed batch fermenter. Now the fourth one is production of organic acid. So here it is useful in the production of lactic acid, and it was first discovered by Scheele. Okay, in 1789. This lactic acid was discovered by this scientist. Sour milk, khatta doodh you say. So from that this lactic acid was produced. The next one is the production of antibiotic. What are antibiotics? Whatever the antibiotics you are taking to treat the disease. Okay, the study of antibiotic began from the discovery of penicillin in 1929. You know that very well. The first antibiotic was penicillin, which was discovered by Alexander Fleming. You know the story very well. I know. I think. Okay, and he proved that broth culture of penicillin notatum has antibacterial property to grow positive bacteria by inhibiting the cell wall synthesis. Most of the penicillin are 6 amino penicillic and acid derivative. So this Alexander Fleming, when he was working on his experiment, he noted that these penicillin antibiotic was having an antibacterial property, okay, to treat the disease. Now you can these microorganisms are also useful for the production of wine. It's like fermentation of fruit juice by yeast. Now commonly in this two types of wines are there, like red wine and white wine. Red wine is generally produced from red grapes, and this white wine is produced from white grapes. Okay. Now the next one is the production of vaccine, who Edward Jenner was the first scientist to produce vaccine. You know very well that smallpox in earlier days, smallpox was used, no, was treated by this cowpox. Okay, cowpox virus. Now this was the production. Okay, now let's see the application of industrial microbiology. What are these applications in these industrial microbiology? Industrial microbiology is one of the largest and most complex of biological sciences, as it, you can say that it deals with many diverse biological disciplines. Okay, and it is also a natural history of microbes. You can use it deals with every aspect of microbe, human and environmental interaction. Okay, now these interactions include ecology, genetics, metabolism, uh, mm, infection, disease, chemotherapy, immunology and agriculture. So these are the applications. Okay, now what is the future of industrial microbiology? Now future challenges are what? The finding new ways to combat disease. Okay, you can find many more ways to combat these diseases. You can reduce pollution, okay, by these microorganisms. There are many microorganisms which are very helpful to our day to day life. Okay, now AIDS, okay, hemorrhagic fever and other infectious diseases. Now you can create new drugs, vaccines, like penicillin was the first drug. In the future, okay, many more drugs can be created to treat the diseases. So use the technique on molecular biology, so create new technique vaccine. You can use this technique in molecular biology and also rDNA to solve the problem. Host pathogen relationship, degrade pollutants and toxic waste, whatever the pollutants are you can degrade it, and also the toxic waste, and it can be used as a vector, okay, to treat the disease and enhance agriculture productivity. So these are the six future applications. What can be the future applications of these microorganisms industrially, especially industrially microorganisms? Okay, so in this in your syllabus, sem 6 syllabus, if you see the sem 6 syllabus, see, so the first topic I have explained is the introduction to industrial microbiology. Now you may get the question, write about the industrial microbiology or any question from this unit. Okay, you can give a small intro about this industrial microbiology, so that your answer can become valuable. Okay, and the examiner can give you the full marks on your answer. So the next topic will be on microorganisms of industrial importance. Okay, what are the microorganisms of industrial importance? Like the in your syllabus, it's yeast, mold, bacteria, and actinomycetes. So I will be explaining in this next video. If you want my videos, so please like my video, share my video, and subscribe my channel, The Learning Hub. Bye bye.